Hi, this is Bithi Ahiri and today's topic is on synaptic transmission. This is part 2. In this discussion, we are going to cover neurotransmitters binding with receptors, message terminating mechanisms, glial and gap junctions, brief overview and the relevant exercises. Our learning objective is to develop a basic idea on synaptic transmission, specially focused on activation of receptor proteins by neurotransmitter molecules and two message terminating mechanisms. Synaptic transmission, especially if we focus onto the chemical synaptic transmission, it includes three broad phases. The release of a neurotransmitter from the presynaptic neuron. That is, we have already discussed in our previous presentation. The last two phases are neurotransmitter binding to specific postsynaptic receptors and the message terminating mechanisms in the synaptic cleft. These two covers will be done in today's presentation. So first, we are going to cover neurotransmitter binding to specific postsynaptic receptors. In this diagram, the presynaptic neuron and the postsynaptic neuron has been mentioned. Here is the dendrite of the postsynaptic neuron. The diagram always helps us to make a clear sense of these topics. So, what happens over here? Activation of receptors by neurotransmitter molecules, that is now we are going to cover. After exocytosis, exocytosis means the releasing of the neurotransmitter from the presynaptic neuron. So after exocytosis, neurotransmitter produce signals in postsynaptic neurons by binding to receptor in postsynaptic membranes. So in the postsynaptic neuron over here, this is the postsynaptic neuron. Over here, the receptor molecules used to be present and with which the neurotransmitter used to mix up or bind up. Thus, neurotransmitter is known as ligand of its receptor. Here in the diagram, the ligand-gated channels has been pointed out here, and the neurotransmitter receptors are already pointed out. Receptor subtypes. The different types of receptors to which a particular neurotransmitter can bind are called receptor subtypes. In this chart, it is given the receptor subtypes are already mentioned in this chart. And for each type of neurotransmitters, there are certain specific kind of receptors which can mix up. This has been given. It was initially assumed that there is only one type of receptor for each neurotransmitter. But this has not proved to be the case. As more receptors have been identified, it has become clear that most neurotransmitters bind to several different types of receptors. Now the types of receptors. This binding influences postsynaptic neuron in either of two ways, depending on the receptor. So receptors are of two types, ionotropic receptors and the metabotropic receptors. In this diagram, it has been shown the ionotropic receptors, which are already known as ligand-gated ion channels, and metabotropic receptors, these are already known as G protein coupled receptors. So here ionotropic receptors, they are associated with ligand activated ion channels. The red circles are in the diagram left hand side. These are the ions, and the neurotransmitters are the green circles. Neurotransmitters binds and the channel opens. This has been shown in the diagram, and the ion flow likewise across the membrane. In metabotropic receptors, they are associated with the signal proteins and G proteins. Here in the right hand side diagram, the G protein is activated, that has been shown, and then the entire process has been described. Ionotropic receptors, this diagram represents the ionotropic receptors. You can see the ligand-gated ion channels are present over here. When neurotransmitter binds with ionotropic receptors, the associated ion channels open or close immediately, thereby inducing an immediate postsynaptic potential. Binding with ionotropic receptor, neurotransmitter brings an immediate postsynaptic potential by ion channels to open or close. Now, metabotropic receptors. This is shown in the diagram that here is the receptors. G protein is present over here. And the second messenger, we will going to cover this topic point also. And the signal molecules are also present over here. Now, metabotropic receptors are more prevalent. Their effects are slower, long-lasting, more diffuse, and more varied. All types of metabotropic receptors attach to a portion of a signal protein outside the neuron. The G protein is attached to the signal protein inside the neuron. Such receptors, when bind with neurotransmitter, G protein breaks, may move inside surface of the membrane and binds with the nearby ion channels, thereby induces an postsynaptic potential. This postsynaptic potential is of two types. One is the excitatory postsynaptic potential and another one is the inhibitory postsynaptic potential. What are they? We will discuss in this presentation later on. Or may trigger the synthesis of a chemical called a second messenger. Second messenger diffuses through the cytoplasm and may influence the activities of neuron in variety ways, may bind to the DNA, influences genetic expression and have radical long-lasting effects. So here we have presented ionotropic and metabotropic receptors. Through ionotropic receptors, fast transmission takes place, and from the metabotropic receptors, slow transmission takes place. Any neurotransmitter can go from the ionotropic, even can go from the metabotropic receptors. So in the left hand side diagram, a given neurotransmitter may interact with many different receptors in different parts of the brain. So here in the left hand side diagram, diagram number A, if we consider, this is diagram number A, a neurotransmitter may activate an ionotropic receptor, and ionotropic receptor also called as ligand-gated ion channel at some synapses, opening an ion channel to affect the postsynaptic cell's membrane potential. The neurotransmitters are pointed over here and the ions are shown. Now, in this diagram, if we consider this diagram as diagram number B, the same neurotransmitter at another synapse activate metabotropic receptors. This is the diagram for metabotropic receptors, which activates second messengers via G proteins that open other ion channels and or cause other changes in the cell. So, in the right hand side diagram, we are coming into the fast versus slow chemical synapses. So, what is the fast chemical transmission? Fast chemical transmission takes place in the ionotropic receptors, shown in the diagram. And slow chemical transmission takes place within the metabotropic receptors with the help of the G protein. This is the ion mechanisms for excitatory postsynaptic potential and inhibitory postsynaptic potential. For excitatory postsynaptic potential, suppose the ions, depending on the ions as well as the neurotransmitters, nature of the receptor molecules, either there occur the excitatory postsynaptic potential or there will occur inhibitory postsynaptic potential. Here I am showing a diagram which help us to make the concept clear. Suppose this is the presynaptic membrane and this is the postsynaptic neuron. In the presynaptic neuron, if the neurotransmitters which are present are excitatory in nature, which are excitatory in nature like acetylcholine, glutamate, or noradrenaline. If these are present over here, it will release into the synaptic cleft, and after releasing in the synaptic cleft, they will mix up with the receptors. These receptors are present in the postsynaptic neuron. These receptors are the proteins which are associated with the ion channels. Each receptors are associated with the ion channels. These are the ion channels. Suppose the ions like Na+, if the ion channels open up and Na+ from the outside comes into the cell, or Ca+, calcium, enters into the cell, or suppose within the cell we know there is the potassium which is positively charged. Suppose this channel for potassium get closed so that the potassium cannot move on. Potassium channels get closed and sodium and calcium channels get open. If these things happen, what will occur? The positive charges will be more within the cell, and that leads to excitatory postsynaptic potential, or in short EPSP. Suppose for inhibitory postsynaptic potential, what happens? This is the presynaptic neuron and this is the postsynaptic neuron. In the presynaptic neuron, the inhibitory kind of neurotransmitters are present. Inhibitory kind of neurotransmitter means we can consider GABA, gamma aminobutyric acid, or glycine as there, suppose. They are releasing from the presynaptic neuron and mixing with the receptors molecules which are present in the postsynaptic neuron. What happens over here? They are associated with the ion channels and the channels will be acted likewise. What happens? Chloride is present, Cn- is present in the outside environment of the cell and supposedly it gets entered. So chloride channel is open and somehow the Na+ as well as Ca+, calcium and sodium ions get closed so that it cannot able to get entered. Na+ and Ca+ cannot able to get entered. These channels are closed. Even the potassium which is at the inside of the cell, the potassium channels get opened. So it comes outside. So what will be the result? The result is the intracellular environment gets more negative some way. So that time the inhibitory postsynaptic potential will be generated. So in this slide, we came to know how the EPSP or IPSP gets formed. Now, postsynaptic potential, also known as graded response. Why? Because the amplitude of EPSP and IPSP are proportional to the intensity of the stimulus that elicits them. So that's why the postsynaptic potential is also known as graded response. Excitatory postsynaptic potential, also known as postsynaptic depolarization, it increases the likelihood that the neuron will fire again by decreasing the resting membrane potential from minus 70 millivolt to minus 65 millivolt. And inhibitory postsynaptic potential, also known as hyperpolarized, it decreases the likelihood that the neuron will fire by increasing the resting membrane potential from minus 70 millivolt to minus 75 millivolt. Here is the diagram. We can see that resting term is mentioned. So here is the amount minus 70 millivolt to be presented. This is the minus 70 millivolt part. So always in such type of diagram, we need to consider that here in the y-axis, there is the amplitude in terms of millivolt. Amplitude in terms of millivolt used to be presented. And in the x-axis, that is the time in terms of millisecond used to be presented. Now the part is that resting membrane potential is minus 70 millivolt. If it goes into the minus 65 millivolt or so, then we can say that it is the EPSP occurring over there. And if it goes into the from minus 70 millivolt to minus 75 millivolt or so, we used to say that inhibitory postsynaptic potential is taking place. Within the neuron, from so many directions, the EPSP and IPSP will be formed. But the sum up things used to be considered whether the next action potential is going to be generated or not. In this portion, minus 65 millivolt is very much important. This is known as threshold of excitation. Many of the times, this threshold of excitation considers from minus 65 to minus 55 millivolt because several neurons have several units to generate the action potential. So once after getting EPSP or IPSP and their sum up things takes place, and once it crosses that limit, we can say that action potential used to generate. So over here in the threshold is written, we can say that either minus 65 millivolt or minus 55 millivolt has been crossed out so that the action potential is generated over there. Through this, we can get the idea that action potential is something which is different from the postsynaptic potential. Now, in this diagram, we are shown that this is the postsynaptic potential. These are all the dendrites. And from different dendrites, the different neurons are associated. So these are the presynaptic inputs. Here is the presynaptic inputs. Another presynaptic inputs is over here. Every synapses is created for everywhere. Like if I found this is portion A, it's a synapse created over here. This is portion B, again a synapse created here. This is part C, another synapse created here. Then part D. So everywhere in every synapse, there are some postsynaptic potentials which are occurring. Some are EPSP excitatory in nature and some are IPSP inhibitory in nature. So excitatory postsynaptic potential is shown through the positive charge, positive sign, and inhibitory postsynaptic potential is shown through the negative sign. Here what happens, after mixing all those potentials, either action potential will be generated. If it crosses the threshold of excitation, either action potential will be generated or it will not generate itself. So action potential used to generate from this region. This is the action hillock region. Here only hillock is written. It's the action hillock region, and this is the initial segment actually, because after that there are the myelin sheaths. From here the myelin sheaths are occurring. These are the myelin sheaths. So from here the action potential used to be generated from the action hillock region. This is the initial segment. So the characteristics of postsynaptic potentials are, it is the graded potential, it's a local response, it is rapid, often it seems to be instantaneous. Their transmission is decremental in nature in terms of amplitude, and it never follows all or none law. So postsynaptic potential across the receptors. Two sorts of receptors we came to know, that one is the ionotropic receptors, another one is the metabotropic receptors. So how the EPSP IPSP looks like in the ionotropic receptors, it's shown in the diagram A, and how the EPSP IPSP looks like in the metabotropic receptors, it is shown in the diagram B. So here I need to mention that there is a concept like synaptic delay. Why? Because neurotransmitter from the presynaptic neuron, it reaches to the postsynaptic neuron. So it takes some time, and the synaptic delay is the time period which is denoted as 0.5 seconds. And the amplitude of each wave is considered to be from 0.5 to 2 millivolt. So that's why here we have shown 2 millivolt in the y-axis part. And the peak of any EPSP curve comes after 11.5 milliseconds. So we can consider that if it is the baseline and if it is the height peak of the curve, then it reaches after 11.5 milliseconds. Here the integration and summations has been focused. Means within the soma, from various direction, the nerve impulses used to reaches. So after that, from various direction it can reach, or various waves can come in a short period of time also. So many neurotransmitters, so many messages are coming, they used to get sum up. The summing up procedure is of two types. One is the spatial summation, another one is the temporal summation. Spatial summation is the sensory summation that involves stimulation of several spatially separated neurons at the same time. Multiple presynaptic neurons are responsible for generating the action potential. Multiple presynaptic neurons generate subthreshold more efficient. This is the more efficient procedure. See here, the simultaneous stimulation by several presynaptic neurons. This is very important. And the presynaptic neuron over here, this is the presynaptic neuron 1, suppose presynaptic neuron 2, presynaptic neuron 3, and from here in all the directions the stimulations are reaches within the postsynaptic neuron, and EPSP spreads from several synapses to axon hillock. Postsynaptic neuron fires from the action hillock region. The action potential used to be generated. In that way it will move again. In this connection, I just want to mention that action potential always goes in the forward direction generally. So if the action potential moves in this way, it is called orthograde action potential. And if the action potential generates, but it starts to flow in the opposite direction, then it is called as retrograde. Retrograde action potential, but it never flows, it never propagates. It used to be over there within the soma, so that the next potentials used to come up, they used to bind up together. And that is the main point, because action potential has its nature to always move in the forward direction. Now into the temporal summation, that involves the addition of single stimuli over a short period of time. A single presynaptic neuron is responsible for generation of action potential. One presynaptic neuron generates subthreshold over a certain period of time, a less efficient process as it takes time to generate action potential. In this diagram, we can see again, this is the postsynaptic neuron. This is the postsynaptic neurons, and so many presynaptic neurons are present. Presynaptic neuron 1, presynaptic neuron 2, presynaptic neuron 3, but only from one neuron the stimulation comes into the soma over here. So here what happens, high frequency stimulation by one presynaptic neuron, EPSPs spread from one synapse to axon hillock, and postsynaptic neuron fires. So over here, one signal at a time used to reaches from the particularly one presynaptic neuron. This is the main important point. Now integration and summation continues. The combination of a number of individual signals into one overall signal is called integration. So spatial summation denotes how local EPSPs of receptive membrane from different locations sum to form a greater EPSP, how simultaneous IPSPs sum to form a greater IPSP, and how simultaneous EPSPs and IPSPs sum to cancel each other. Secondly, in the temporal summation, it denotes how postsynaptic potential produced in rapid succession at the same synapse sum to form a greater signal. So here is the diagram presented. This particular portion will be more clear if we focus into this diagram. Here, diagram A, no summation is taking place. So this is the minus 70 millivolt that denotes the resting membrane potential, and this is minus 55 millivolt means that action potential will generate after this amount. So what happens? Two stimuli separated in time cause EPSPs that do not add together, and from here no action potential is going to be generated. In diagram B, this is the temporal summation. Temporal summation, what happens? Two excitatory stimuli close in time cause EPSPs that add together. That is very important, that add up together, and it crosses minus 55 millivolt. So over here the action potential has been generated. Then in diagram C, this point is very important. Two simultaneous stimuli at different locations cause EPSPs that add together. Here is the, in diagram C, here is the E1 that denotes presynaptic neuron 1. From here the messages used to come. Again, this is E2, that is the presynaptic neuron 2. From here also another message come, but two messages come simultaneously, so that they binds up together. So EPSP are add up together over here. Two excitatory signals are coming from two different part, and they add up together. The addition voltage crosses minus 55 millivolt and action potential generates. Comparing to diagram B, if we compare with diagram B with diagram C, we will find in diagram B one excitatory solitary presynaptic neuron is over there, and from that direction only two stimulations comes up. So these two stimulations are close in time respect, but they are coming from the same presynaptic neuron. Now for the diagram D, spatial summation of EPSPs and IPSPs, what happened? E1 denotes that presynaptic neuron, that is the first presynaptic neuron, that is excitatory in nature. But the second one, I1 presynaptic neuron, that is inhibitory in nature. So E1 and I1 from two presynaptic neuron, from where first one the excitatory signal is coming down, and from the inhibitory presynaptic neuron the inhibitory synaptic signal is coming up. They club up together, and ultimately what happens by climbing up, it is not able to reach minus 55 millivolt. So there is no action potential going to be generated. Now message terminating mechanisms in the synaptic cleft. Here two mechanisms of deactivating neurotransmitters that terminate synaptic messages and keep that from happening are reuptake. It is often occurring. Reuptake is very important. Majority of the times we can find that reuptake is taking place. Often neurotransmitters once released are almost immediately drawn back into the presynaptic buttons. So that is called the reuptake. And from enzymatic degradation, neurotransmitters may be degraded or broken apart in the synapse by the action of enzymes. So here in the diagram, the enzyme has been pointed out, as well as the reuptake process has been shown over here when the neurotransmitters are going back. This part in the diagram, it is shown that inward direction towards the presynaptic neuron, the neurotransmitters are again moving. That is the returning back of the presynaptic neuron. That process is the reuptake. Glial function in the synaptic transmission, especially the astrocytes, release chemical transmitters to contain receptors for the neurotransmitters, to conduct signals and to participate in neurotransmitter reuptake. So glia is very important in this regard. Nowadays, so many researches are taking place. Gap junction, another important point. This diagram, if you carefully look onto the diagram, you will find that in the electrical synapse, that is B, diagram B, there is the presence of gap junction. So gap junctions are narrow spaces between adjacent neurons bridged by fine tubular channels that contain cytoplasm. Cytoplasm allowing electrical signals and small molecules to pass easily from neuron to next neuron. Gap junctions are often called the electrical synapses, and it is very rarely found in mammals. So this is the steps of synaptic transmission and the diagram taken from the Encyclopedia of Britannica. Here in this presentation, we are actually covering the process which is taking place after neurotransmitter has been released from the presynaptic neuron. So in the first point, it has been mentioned action potential reaches to presynaptic terminal. Secondly, depolarization of the presynaptic terminal opens ion channels, allowing calcium into the cell. Calcium triggers release the neurotransmitter from vesicles. Point number 4, neurotransmitters binds to receptor sites on postsynaptic membrane. In this presentation, we are starting from here because in part 2, we have this is the part 2 of synaptic transmission discussion. In the part 1, we have already covered the first 3 points. So from here, point number 4, we have started our discussion. And in the 5th, opening and closing of channels cause change in postsynaptic membrane potential, and action potential propagates through the next cell. Neurotransmitter is inactivated or transported back to the presynaptic terminals that we have covered over here. So in the brief overview, if we focus that what are the things in today's discussion we have covered. We have covered neurotransmitter binding to specific postsynaptic receptors. And under this, we have already discussed activation of receptors by neurotransmitter molecules, receptor subtypes, postsynaptic potentials, and summation or integration part. Then the message terminating mechanisms in the synaptic cleft, and within this reuptake and enzymatic degradation has been discussed. And after that, 2 points has been covered, that is the glial function in synaptic transmission and gap junction. So in the exercise, you need to cover, you can able to answer these points, discuss the synaptic transmission of nerve impulse in detail, distinguish between EPSP and IPSP, postsynaptic potential and action potential, ionotropic and metabotropic receptors. Then you can able to write short note on ligand and integration in synapse, role of dendrites in synaptic transmission, role of glial in synaptic transmission. If within this discussion and in this question, if you found any difficulty, you can mention me in the comment section also. And if your concepts get more clear or you can understand the presentation properly, if it is helpful, then you can subscribe the channel, share it with your friends, and finally, thank you for your patience.